Welcome to an introductory video tutorial on the mammalian food entrainable oscillator. In this video, we will discuss what a food entrainable oscillator is, how it was discovered, and what we know about where it's located and how it works. Light is an important synchronizer of circadian rhythms through the suprachiasmatic nucleus, or SEN. However, in conditions where food availability is limited, the feeding schedule can also regulate circadian rhythms, leading to the idea of a food entrainable oscillator, or FEO. Review the evidence that led to the discovery of an FEO that controls behavioral rhythms, and demonstration that it is separate from the SEN. Here is a model actogram from a rat in a 12-hour light-dark cycle. In the first part of the experiment, food is available ad libitum, which means it's freely available. Since rats are nocturnal, they are active during the nighttime. After a couple of days, the food is removed. Rats can tolerate short periods of food restriction without ill effect. Food restriction alone does not substantially affect the locomotor rhythm. After that, the food is available for only 2 hours when the lights are on, when rats are usually inactive. The time of food availability is represented by the blue rectangle. We can observe that, after the first few days of food restriction, the rat shows high levels of locomotor activity for approximately 1 to 4 hours before the time the food becomes available. This is called food anticipatory activity, or FAA. To show FAA, the animals must be exposed to a restricted feeding schedule to be able to predict when the food is going to be available, and to anticipate it in the absence of any other cue. For example, the rat does not show FAA on the first day of food restriction, and the FAA arises only after the restriction pattern is established. Sometimes the animals show less intense activity at the end of the night after FAA, indicated by the fading activity bars. After FAA is established during several days of restricted feeding, the food is removed entirely for 3 days. As you can see, the FAA still occurs, anticipating when the food used to be available. If the FAA were a homeostatic hunger response, we might expect the rat to have increasing FAA behavior with every hour since it last ate. Instead, the FAA has discrete occurrence around the time of previous food availability. This demonstrates that FAA is not due to the presence of the stimulus, in this case, food, but rather is controlled by an oscillator that is entrained to food availability and continues to keep time after the food is removed. Next, the food is provided ad libitum again. FAA disappears quickly and the rat resumes its usual locomotor pattern. 3 days later, food is removed again, and the FAA comes back at the same time as it occurred before. This allows us to verify that the phase is still controlled after the release in the food-restricted condition. Even after 3 days of unlimited food availability, the FAA appears again as soon as food is removed. These findings suggested that an entrained oscillator underlines FAA, hence the use of the terminology food. However, because these experiments were done in light-dark conditions, it is impossible to know if the FAA is a part of the light entrainable oscillator or is controlled by some other independent oscillator. This next experiment addresses the contributions of light and the SEN. It is a model actogram from a rat in constant darkness and with a lesioned SEN. In the first days, the food is available ad libitum. The rat is arrhythmic under these conditions due to the SEN lesion. When the food availability is restricted, the rat shows FAA prior to food availability, just like the intact rat before. The fact that the SEN lesion rat still exhibits FAA in constant darkness means that the light entrainment and the SEN are not required for the FEO to drive FAA. Next, the experimenters delay food access by 2 hours. The rat quickly entrains to the imposed cycle again and does so in the absence of any other cue. The period of the rhythm clearly matches the period of food availability, when food is available approximately every 24 hours. We don't show it here, but if food is available in a non-circadian pattern, such as every 18 hours, it will not entrain. We can also tell that the phase relationship is repeatable, because the onset of FAA always occurs a set period of time before the food is available, regardless of when the food is presented. Next, the experimenters delay food access by 8 hours. Here, activity is first seen when the food used to be available, but only persists for a few hours and stops prior to food availability. Again, if FAA were simply a function of time since last feeding, we would expect the activity to persist until food was available. However, it does not, and instead slowly drifts to the time of new food availability until the rhythm becomes entrained again. Altogether, the results of these two experiments demonstrate that FAA is not the result of masking or hunger, but of an oscillatory system that can be entrained by a fixed time of food availability, and that is independent of the SCN or photic input. We know that many mammalian circadian oscillations are mediated by a transcription-translation feedback loop of the canonical clock genes. Mutations of the period and cryptochrome genes can alter the period of FAA expression, suggesting that the molecular circadian clock contributes to the oscillation that drives FAA. However, multiple studies show that FAA persists in arrhythmic rodent models, including the clock delta 19 and BMAL1 knockout mutants. These mutant animals, the FAA persists in light-dark conditions, and also during fasting and constant darkness, demonstrating that neither the SCN nor canonical clock genes are necessary for FAA. Because we know the SCN is not required for this FEO to function, a lot of research has looked for the anatomical basis of the FEO, but there is no consensus. Some believe that it is not localized to a specific tissue, but is a widely distributed network where no single part is essential, which would explain why it is so hard to find. In this case, lesion of a single area would not be sufficient to block FAA. Let's review how the locomotor FEO meets the properties of a circadian oscillator. To test constant conditions, food would need to be restricted long enough to see a free-running rhythm of the FAA, and it is not humane or practical to stop rodents for a prolonged period. We cannot easily test this property of the FEO. The period of FEO matches the Zeitgeber. FAA follows 24-hour cycles of food availability, except when it is re-entraining after a phase shift in the feeding schedule. The FEO has a stable phase relationship with the Zeitgeber, because stable FAA starts a set number of hours before food availability each day. The phase of the FEO is controlled after releasing to constant conditions when FAA reappears during complete food restriction and maintains the same phase as with the original feeding cue. We don't know if the FEO is temperature compensated. If we knew where the FEO is located, we could evaluate temperature compensation with tissue culture experiments. We may never know if the FEO satisfies all of the properties of a circadian oscillator. However, scientists have shown that it has oscillator-like properties that make it interesting to study.